Okay, aloha everyone. My name is Ryan Feroy. I am the chair of Erica's thesis committee and also chair of the Department of Geography and Environmental Science and the director of the Spatial Data Analysis and Visualization Lab. And it is my, true honor and pleasure to introduce Erica this morning, November 16th, 2021. For her thesis defense in the Department of University of Hawai'i Hilo Tropical Conservation and PC Biology and Environmental Science program. So Erica has been with us for a few years now. We're really just so happy to have her work in the lab and the contributions she's been able to make across all kinds of different projects. If I had a virtual lei, I would put it on Erica. If we were in person, she'd be covered in lei. But we're doing this remotely, which is, I think, excellent opportunities for some folks who might be far away to participate in this and see the great work that Erica's been doing. And I think that Erica, more than almost any of my other students, has really promoted an interdisciplinary project in the true sense of the word, where she's marrying sort of, ecological, environmental science concerns with what we can, learn from the power of, machine learning and artificial intelligence and data science. And so Erica's project has really brought those together in a way that none of my other prior students have had an opportunity or been able to do. And so that's really exciting. I think her work is really pushing us forward in what we can do for thinking about invasive plants in Hawaii and beyond. And so quite excited for Erica's talk. And I'm not stalling. Honestly, I have not been stalling, but the last committee member has just joined in. So this is a fantastic opportunity for me to now hand the floor over to Erica for her presentation. And I hope you all enjoy it very much. And at the end of her presentation, we will have some time for public discussion and public questions for Erica. And then after that, we'll ask all the public to maybe go about your rest of your day, and then the committee will be talking to Erica after that. So that's the plan. And with that, I'll mute myself and hand it over to Erica.
Thanks, Ryan, for that introduction. Happy to be here, and aloha and good morning, everyone. Again, first wanted to thank everyone for showing up this morning. I know it's pretty early, especially for those in Hawaii time. And my name is Erica, and I'm happy to be sharing my research with you on invasive species detection from RGB aerial imagery, where I identify links between plant characteristics and transfer learning methods. And for those who are unfamiliar with the acronym RGB, it stands for red, green, blue, and in the context of imagery refers to true color images. And to start off, just an FYI, I'm going to go complete 180 and talk about something that's very different from the terrestrial environment. I've noticed some parallels between my research and one of my favorite hobbies. So when my friends and I get the itch to dive, the first question is always where. We never really debate whether we should dive, but where we should dive. And when we all consider a place to dive, we think about which place is going to offer us the best conditions to thrive in our session. And that's going to be ideally no wind, no swell, no tiger sharks that we know of, and fish, of course. And a similar concept occurs when a species gets an opportunity to grow in a location outside of their native distribution. And when they're in an area with no natural predators, a mild climate, and abundant energy sources, species can live their best lives elsewhere. And it's unfortunate, though, because these species can grow uncontrollably in these environments and as a result disrupt the existing native ecosystems. So they're harmful not because of what they are, but where they are. And invasive species in Hawaii is pretty apparent because of their damaging effects. And to elaborate, native ecosystems are likely undergoing changes in biodiversity, biogeochemical heterogeneity, and ecosystem services due to occupation of ecosystem modifying invasive species. And my research actually focuses on invasive plants versus invasive animals. So this is a map showing the top 17 ecosystem modifying invasive plant species and their locations across Hawaii. And this is just showing the same list or legend we just saw from the previous map. And my study focuses on two of these species. One is a grass, which is now formally known as Panica maximum, and another plant called Myconia calvescens. And this is a photo of what the grass looks like. It's now Megathrysis maximus, and it's commonly known as guinea grass. And here is Myconia. Both plants, which you've probably seen before, definitely in Hilo. And the third invasive species that's part of my study is another very prominent one here in Hawaii, and it's an invasive fungus. There are two fungi, Ceratocystis leuco-Ohi'a and Ceratocystis huli-Ohi'a, and they're collectively known as rapid Ohi'a death. And these two fungal pathogens are actively attacking and killing Ohi'a trees. And this is a big concern because Ohi'a is a foundational species of Hawaiian forests, and rot has the potential to cause a lot of ecosystem disturbances that negatively impact watersheds, cultural traditions, natural resources, and quality of life. And I wanted to revert back to this map because we can see that these invasive species cover, and these invasive plants cover a large surface area across the islands. And monitoring and tracking these invasive plants can be done in different ways. But a really prominent one and useful one is remote sensing. And remote sensing is able to acquire information about an object without making physical contact with that object. So a popular source of remote sensing is using satellite imagery. But the problem with satellite imagery in our case is that these plants that we're interested in are too cryptic and they're too small to be seen in most satellite imagery. So in our lab and for our purposes, we primarily deploy drones and different tools that give us high resolution imagery so we can see plants much better than we can using satellite imagery. And as I mentioned before, in SDAV Lab, we use drones, and we also use helicopters to collect our imagery and collect our data. And for the helicopters, we use this cool rig that we can attach to the belly of a helicopter. And on that rig, we can attach GoPros and DSLR cameras. And for context, with drones, we can cover hundreds of acres and collect data across hundreds of acres. And for helicopters, an advantage is that we can collect thousands of acres. So this gives us a lot of data to collect on the suite of plants that we survey for. And I always think these videos are fun. It's cool to see what the helicopter sees. And this is a rod survey that's happening down in the Kahuku unit in Hawaii Volcanoes National Park back in 2018. So this is cool. After one of these helicopter and drone surveys, we're able to collect kilometers of imagery for our study sites. And this is rod, for example. And you can see, the extent of our study site. And here's an example of one of the images we collected from one of these surveys. And it's clear that you can tell that there are rod suspect trees within them. So that's good news. These high resolution images work really well. And take Guinea grass survey, for example, we can collect tons of data within its study site. And this is an example of one of the high resolution images we can grab. And I forgot to mention, both of these images were taken from the helicopter rig setup and captured with DSLR cameras. And so this is awesome. We have all this imagery and tons of data, but it's actually a really big challenge to analyze this many photos. It's like combined, it's 1,500 plus photos. And that's a lot of photos for someone and a human to just go through and manually pick out one by one where our invasive species targets are. So we need a better way and a systematic way to process all these photos more efficiently. So enter artificial intelligence. And artificial intelligence leverages computers and machines to mimic the problem solving and decision-making capabilities of the human mind. And under the umbrella of artificial intelligence is machine learning, and machine learning uses algorithms to identify patterns within data to create a model that makes predictions. And to elaborate even further, deep learning, which we'll be focusing on my study, is it's known as a scalable machine learning. And it requires less or no human intervention to process data. It can ingest unstructured data in its raw form, such as text or images, and it can automatically determine features that distinguish different categories of data. And one of the most popular applications of artificial intelligence these days is computer vision. And computer vision is a field that involves how computers gain a high-level understanding from digital images or videos, and that seeks to automate tasks that the human visual system can do. And there are many applications of computer vision, but these are arguably the top 3 most common ones. And the first is classification on the left, where a photo is simply labeled by what the photo entails, and object detection, where objects of interest are identified by what they are and where they are located in a photo. The third is image segmentation and the process of portioning an image into multiple segments. And for this study specifically, we're focused on object detection because we can identify and we can extract geographic locations of plants. So some examples of deep learning being used in the ecology space include identifying tree crowns in a landscape, or here where we're looking at palm trees that are circled in red. And then here, an image segmentation example where coral structures are segmented to classify boundaries of coral, water, and sand. And in deep learning, building and training a model entirely from scratch requires a great amount of labeled data, also known as annotations, big clusters of computer servers and a lot of time. So it's very resource intensive if you're trying to do this from scratch. So an approach to this is transfer learning. And instead of training from scratch, transfer learning is a method used in deep learning when sufficient labeled training sets are unavailable. And it refers to a process where a model trained on one problem is used in some way on a second but related problem. And in SDAV Lab, given our smaller data sets in comparison to a lot of data sets that are used in deep learning problems, we have always utilized transfer learning in our methods for this reason. And how transfer learning is typically used, it involves training a model on a large source data set and taking that information from that model to help train a task that has a much smaller data set. And overall, it's good to note here that there isn't really a guide to transfer learning or a one size fits all or a silver bullet method to produce any ideal outcome when using transfer learning. So as I mentioned, a common transfer learning method is to use a pre-trained model from one of these large data sets and apply it to a task of choice. And large data sets include stuff like MNIST, there are these photos on the left, MNIST, Pascal VOC, and ImageNet. And all of these are popular data sets that have up to like millions of annotations across hundreds of different categories. And in our object detection workflow, our lab uses ImageNet, but the downside is that all of these images found in any of these data sets often look completely different to the plant data we collect, especially the plant data we collect in aerial images. Like if you just take some examples from what you can see even in like Pascal VOC, it's just of birds and dogs and buses. And none of those things really look like Myconia or guinea grass. And even now and for future use cases, data limitation is likely going to be a constant hurdle. There is no exact number or how much data is needed to produce an accurate detection model on a plant by plant basis. And although it'd be possible to collect more data if we needed it, continuously collecting imagery and creating annotations is pretty costly. And we aren't able to turn to the internet in our case because, or turn to the internet to look for more data because in our case, certain plants we're interested in are only found here in Hawaii and can be pretty rare. And like we saw before, data sets like ImageNet are not enough to solve these kinds of problems. So we need to bring in other data sources. And in addition to having data limitations, we're also looking for a more efficient system to ID plants in our data, in our imagery. And first off, a lot of human effort is needed to create annotations or to identify targets in an image. And second, we have a lot of data sets from all the surveys we've done, but we don't know the best way to maximize these data. And without knowing how to utilize the data we have, continuously collecting data will continue to be expensive. So ultimately, our hopes and dreams are to create a system that could tell us which data is most useful to bring in to help improve performance. And going back to the topic of deep learning for a little bit, I wanted to share things we know and what we don't know and what's ambiguous in deep learning. So deep learning is highly capable of distinguishing features automatically in images, but how features are mapped and how they're recognized and how these algorithms learn these features from images is not very human interpretable. And here are some examples of that. You can see the feature maps are in blue and the source images are on top. And if you took away the source images, I would not be able to tell you what was going on in the blue images. And this is another example of feature maps from an image. So we know that features are playing a role in deep learning, but we don't know which features are influencing a model's detection performance. However, what's intriguing is that we can easily look at explicit features that are commonly used to interpret aerial images to help us kind of hone in on how well an algorithm can detect plants. And quantifying these image features in aerial imagery could suggest which data is most useful to apply deep learning and where it is most useful to transfer. And some of these explicit features include things like shape, noticing the shape like here, and where these meandering rivers show like U shapes. Sizes, this one's looking at size of highways, like a residential road versus a multi-lane highway. And tone and color, where we're seeing these contrasting scenes within an image. And texture, where this one's kind of looking at the roughness of a forest and then the smoothness of water. And because transfer learning is such a big part of the study, I wanted to emphasize the method of transfer learning we're using, with the end goal of figuring out how data sets can help improve object detection performance. And while most studies just use a single level of transfer learning, this study uses a second level of transfer learning. So to explain, we first start off with standard transfer learning method, where we take knowledge, or we take information from ImageNet, and we use it to train a smaller task with a smaller data set. And for example, this would be our plant number 1 target with a smaller source data set. And then for the second level, we're going to take this same plant number 1 source model, and we're going to apply that information to a plant number 2 target with an even smaller data set. So multiple levels of transfer learning going on. And keeping in mind all these image features and transfer learning methods, studying the roles they play could be a way to connect these two worlds of invasive species management in Hawaii and object detection techniques. And we first want to know how plant features influence object detection performance. And second, how can we leverage our existing plant data for transfer learning? And more explicitly, my first research question is there any relationship between a plant's object detection performance and its contrast, shape, size, or texture features in imagery? And second, which species significantly improves detection accuracy most after cross-species transfer learning? And third, more broadly, how much would transfer learning help in a situation where both the target both the source and target tasks are of plants, versus before we were seeing, everyday objects being transferred to plants, where in this case, it's plant to plant. Moving on to my methods, I first wanted to just formally show my study species. First is guinea grass on the left, and Myconia in the middle, and rapid Ohi'a death on the right. And for rapid Ohi'a death, actually, we split it up into 4 symptomatic stages. And red is on the far left, and this is the first signs of rod. And brown is the stage where trees begin to lose color and their leaves. Fine white is a loss of leaves, but a lot of the fine branches remain. And the final stage is skeleton, where there's pretty much a complete loss of leaves and fine branches. And at a quick glance, these plants look pretty similar, especially red, brown, and fine white. And the motivation for us including these classes was to see how good improvements are when the source and target are quite similar to one another, such as red rod or brown rod, versus when they're pretty different, like if we were comparing red rod to Myconia. And here is just a brief overview of the components of my methods. The first part is the feature measurement portion, where I'm collecting measures of contrast, shape, size, and texture per class. The second is the object detection portion, or the deep learning portion. And the third is the transfer learning portion, where the end goal is to compare a model that uses a significantly less number of annotations and compare that performance with something that has the same amount of annotations, but the information of a previous model. And here are our feature measurements. So we have contrast, which is going to be the index of root mean square. Contrast, shape, which we're using circularity as our index. Size will be measured in square pixels, and texture is measuring the variance. And here are some visualizations of contrast. And it's pretty cool. This graphic shows different contrast measures from different parts of a photo. And the area that has highest contrast is shown at the very bottom circle below at 0.337. Here is an example of contrast measure, or sorry, circularity measure. And we can see the higher the roundness, the higher the circularity measure will be, the lower or more like jagged or more edges it has will be lower, will be a lower circularity measure. And for size, we can see that it's in, we're seeing how many square pixels an object takes up in an image. And this is texture. So to collect all of these feature measurements, I used ImageJ, and ImageJ is an open source image processing program. I collected measurements for my 6 objects of interest for guinea grass, Myconia, red rod, brown rod, fine white, and skeleton. And I collected 50 samples per object. And this is just to quickly show some model parameters. I won't go into more detail about them, but if you have questions, please hold them. I will definitely talk about these if you'd like. This is our general object detection framework. A lot of these components can be thought of as building blocks, but for our purposes and the architecture we use Keras as our API and TensorFlow as our open source machine learning platform, RetinaNet as our object detection model, and Backbone as our, and ResNet 50 as our backbone. And this slide has some important information of the model experimental design of my study. So we created these tiers of models to compare any improvements or decreases from models that use transfer learning and models that do not use transfer learning. So because the focus is transfer learning, once again, our bigger data set has 500 annotations, which is our source data set, and our target data set, as well as transfer learning data set, only has 25 annotations. And the reasons for 500 is because across all 6 of my objects of interest, they all had at least 500 annotations. And I wanted to be able to compare. So I just decided that all objects should use 500. And the reason for 25 annotations for smaller data sets of target and transfer learning was to simulate an object of interest that had a severe data limitation. And in general, it's good to note here that the number of annotations are low, especially in comparison to studies that use a minimum of 1,000 annotations per class. But the focus of the study was to see any performances or any performance increases or decreases rather than produce a good object detection model. And this is a super abridged, simplistic workflow of object detection where we choose a target, we do some image prep and data prep on our images, and then we run all that data through our algorithm, and we're output with new predictions on new data. So the labeled data part I was talking about refers to this. So this is an example image where we are labeling and drawing bounding boxes around our target of interest. And in this case, one photo has red rod, brown rod, and fine white rod. And this image software is called Labellmg. And here I describe or illustrate more of what's going on in the object detection workflow. So, right, we start off with a target of interest, and in this case, I chose my Konya. And we're gonna do some data prep on there where we take our full-size image, segment that down into 1,000 by 1,000 pixel sizes. And then on that 1,000 by 1,000 pixel sized image, we can then draw our bounding boxes and make our annotations on that photo. And once all of our annotations are ready, we can input all of them into our deep learning algorithm. And what is output is predictions of where our targets are in a photo. And this is the transfer learning component where, again, this is the first level. So we're taking information after learning from our ImageNet data set, and we're applying it to learn guinea grass, for example. And we're using this same method and applying it to all 6 targets of interest and using 500 annotations to learn them. And this gives us our source models per object of interest. And now the second level transfer learning, we take our source models that we just created in the previous slide, and we apply that information to another class. That's one of our 6 classes of interest. And then we compare the performance of this transfer learning model to a target model that does not have a transfer learning component. And it's the transfer learning model in yellow and the target model in green are 2 separate models, and they don't interact with each other, whereas the source model in blue is sharing information with the transfer learning model in yellow. And this is just to show that in the end, we're going to compare model performance between transfer learning and model performance that does not have transfer learning. And to give a more concrete example of what I mean by the second level transfer learning, we'll take our source model that we just made of Myconia using 500 annotations, and then we're going to apply it to help learn fine white rod using just 25 annotations. And so then we're going to compare the model performance of our transfer learning model to a fine white model that does not have the transfer learning component or the information of Myconia. And this is just showing more of the experimental design of the model component. And a lot of variation occurs when you are training these models. And to account for that variation, I ran each model 5 times each. So in the target series component, all the models are run 5 times each per class. And to explain the source and transfer learning models, I did the same thing. Each class's model is run 5 times each. And for the transfer learning models, this is where it gets a little more confusing, but this table is illustrating the matrix of transfer learning instances across all of our 6 classes. So setting it up in this table is just helpful to see and keep track of what class is getting applied to which target class. And again, for all of these transfer learning instances, I ran them 5 times each. But what will be populated in this table is the difference in model performance of guinea grass to Myconia, for example, versus just Myconia trained with 25 annotations. So we're taking our transfer learning model performance, which here for example, would be guinea grass to Myconia. And then what will be populated is that model minus the performance of the target model. And in the final prediction output, we end up with bounding boxes that represent ground truth boxes seen in blue and prediction boxes seen in yellow. And how prediction and ground truth boxes get matched up is by something called intersection over union and or IOU. And IOU decides whether a prediction is correct with respect to an object or not, and is defined by the intersection between the predicted bounding box and the ground truth bounding box. And for this study, IOU was held at 0.5, meaning there needed to be at least a 50% overlap for a prediction to be considered correct. And if there was a prediction less than, or if there was a prediction and a ground truth that did not have at least 50% overlap, then it's considered a false positive. And here, what's actually output as well in these predictions is how confident these predictions are. So you can set confidence thresholds where in this photo, the confidence threshold is 15%. And you can see that every prediction, which is every yellow bounding box, has a prediction that's at least 15% confident that this is the target. And in this photo, the confidence threshold changes to 40%. And with a higher confidence threshold, you'll get less bounding boxes that exist, or less predictions that exist in a photo. And to summarize the performance metrics that I used in this study, the output metric is called mean average precision. And this metric averages all confidence thresholds at a single IOU threshold. And this integrates the entire area under this curve you see here. And what's on this curve is precision and recall. And precision is, in essence, the true positive predictions over all predictions, and recall is the true positives over all of the ground truth. And for our results, I wanted to first start off with the feature measurement portion. Over here, we can see that kind of similarities emerge between or across our classes of interest. So in rod, for example, in size, red, brown, fine white, and skeleton measure pretty similarly in size versus guinea grass and Myconia measure similarly in size. And for circularity, red, brown, and fine white measure pretty similarly in circularity, whereas skeleton and guinea grass also measure similarly And this is just to show that by looking at some of these features, we can tell and we can pick up on certain patterns. And correlations were ran to answer whether there is any relationship between model performance and contrast, circularity, size and texture. And he found that contrast is significantly correlated with model performance, and texture is almost significant, but it is strongly correlated. But circularity and size don't seem to have any relation to model performance. So the results of the cross-species transfer learning are in this table. And again, it is the difference in transfer learning model performance and the target model performance that does not have transfer learning. So in essence, a positive value means that transfer learning helped in this case. And specifically, or more interestingly, the boxes that are green and bolded are significant increases in model performance. And the significance refers to just p-values less than Bonferroni adjusted alpha level of 0.00167. And we can see in this photo or in this graphic that gains mostly happened between rod classes in this section right here. So to discuss this table in more detail, 9 out of 30 instances significantly increased performance, whereas within those 9, 7 instances increased by at least 0.1. And when the source and target classes were the same and of rod classes, 6 out of 12 instances significantly increased performance. Whereas when a source class was a non-rod and the target class was a rod, the only instance where performance increased was between skeleton beginning grass. So it's interesting that gains mostly happened when it was between rod species or rod classes, whereas there was pretty much no gains otherwise. And I wanted to just visualize the overlap when we're plotting our feature variables. So with variables of size, texture, and contrast, our classes like brown and seen here in purple, fine white seen here in pink, and skeleton in green, they're showing the most overlap. So it's pretty interesting that just visually you can confirm like there's some kind of similarity going on. In summary, we learned that given small data sets, standard pre-training from ImageNet did better than models that underwent a second level of transfer learning, but models that had 2 levels of transfer learning had most gains when it was between rod classes. And as far as our feature measurements go, contrast was significantly correlated with model performance. And how this can be useful for the future is that we can think of transfer learning as something that can be helpful, but it's not going to be a one size fits all for all species. And where we choose to use transfer learning should be used with discretion. And to help decide which features or which data sets we can use in transfer learning, feature measurements like contrast and texture may be helpful to determine that. And I want to leave you with an A new species of interest we're interested in, and that is of interest to Hawaii Volcanoes National Park, is Morella Faya. And what I did here was I took some measurements on some Morella Faya data and compared it to future measurements of all of the other classes in the study. And it most similarly measures up with the rod classes. So potentially Faya could be of use when we're transfer learning from rod classes. And to see where, Faya trees contrast and texture kind of measure, we'd expect that with Faya's contrast of 45, maybe its model performance would end up somewhere up here. And for texture, maybe the model performance would end up somewhere around 0.5. And with that, mahalo. Please share any questions or comments you have with me.